It is my great pleasure to introduce Dr. Janusz, Professor of Pediatrics, Experimental Medicine, Human Genetics and Biochemistry, and the Jack Cole Chair in Pediatric Hematology Oncology at McGill University. He is also an investigator at the Research Institute of the McGill University Health Center in Montreal, Canada. His laboratory investigates how oncogenic events deregulate vascular tumor microenvironment, orchestrate intercellular communications, and impact processes mediated by the exchange of extracellular vesicles, including exosomes. He uncovered extracellular vesicles carrying oncogenic cargo, also known as oncosomes, an area in which his group is developing diagnostic applications such as liquid biopsies in cancers across the different age spectrum in patients. Recently, these studies have increasingly concentrated on pediatric brain tumors. He's also among the founding members of the International Society for Extracellular Vesicles, and he also leads the NET program at the Research Institute of McGill University Health Center. His talk today is titled The Rise of Exosomes, the Emerging Role of Extracellular Vesicles and Particles in Liquid Biopsy of Cancer in Children. Today, he'll outline to us the role of liquid biopsy in cancer and the emerging role of this approach in pediatric malignancies. So, without any further delay, I hand over the webinar to Dr. Janusz. Thank you.
Well, thank you. Thank you for this very kind introduction and for inviting me to this webinar with the organization that's legendary for support for pediatric research. And I'm, of course, very honored to be a part of this event. And so what I'd like to tell you about today is, as mentioned, the title is The Rise of Exosome, the Emerging Role of Extracellular Vesicles and Particles in Liquid Biopsy. And this information that I'll try and convey and discuss with you comes partly from our own work, which is sort of a contribution of a group of people who work with me, as well as collaborators and supporters of our work through different funding mechanisms, as shown in these slides, as well as large parts of the content will come from the literature, simply because there's a growing field of liquid biopsy researchers, and they deserve to be acknowledged, and their work is, of course, incredibly important. So my first sort of comment that I wanted to sort of begin with is the cancer is fundamentally a systemic disease, and I think this is not always appreciated, especially in view of some of the cancers being localized. For example, pediatric brain tumors tend to be localized, but we know that this is a systemic condition almost from the beginning, because there's a number of systemic manifestations, be it remote responses or paraneoplastic syndromes, that are frequently associated with cancer progression. This could be, for example, immunosuppression or immunostimulation, perturbation in the hemostatic system, some neurologic symptoms as shown here, endocrine and metabolic syndromes, dermatological and otherwise. And of course, the culmination of these processes is formation of pre-metastatic lesions, different organ sites, and metastases. And while we reflect on these systemic manifestations, what do they mean? It's essentially inescapable that the fundamental property of cancer cells must be a release of some of the mediators of these effects into common fluid spaces, for example, such as blood or other biofluids. And we already know that there's a number of entities or molecular or non-molecular entities that can be detected in circulation of cancer patients using different techniques. So, for example, there's many molecular entities such as hormones or cytokines, chemokines, and so on. But there's also a class of material released from cancer cells, which have certain cancer-specific properties, and those include circulating tumor RNA, for example, or material contained in tumor-educated platelets, as well as circulating tumor DNA, genomic DNA coming from cancer cells, as well as cancer cells themselves, circulating tumor cells that can be detected in the peripheral blood of cancer patients. And finally, there are these entities that have relatively recently come to the picture, and they include extracellular vesicles and particles, which are essentially cell fragments that enter biofluids, coming out of tumor mass and can be detected and analyzed. And the message here, I suppose, is that blood therefore may contain vital diagnostic or biological information about the underlying cancer, which is, of course, accessible and can be tapped into for a variety of purposes. And I suppose this notion is at the very foundation of the promise of liquid biopsy. And many of you, of course, know that this notion is essentially encapsulated in this idea that by testing different biofluids in cancer patients, one can non-invasively, repetitively, in real time, with relative accuracy, detect cancer or detect many of these properties of cancer under investigation, and this may lead to certain profound realignments in our thinking about the disease as such. For example, we know that in many cases, unfortunately, cancers do relapse or become resistant to therapy, and the clinical detection of these stages of disease progression come relatively late. They're usually based on imaging or clinical signs. And yet, if it was possible to detect these processes ahead of time using liquid biopsy approaches, one could gain valuable time and use this time toward many decisions that would profoundly affect the course of disease and patients' well-being. So this could include, for example, early clinical diagnosis or prognostication, disease monitoring, minimal residual disease detection, drug resistance, and so on. So this is the idea of liquid biopsy, which I'm sure everybody knows. And the question is, does this actually work? Does this idea actually work in real life? And I'd like to, if we had this conversation, for example, or this discussion, maybe 5 or 10 years ago, I would probably be, hand waving and pointing to some future benefits in a very sort of theoretical and experimental way. But in 2024, the future is now, and this is reflected in some of the literature that is populating the field these days. So, for example, some of the very sort of methodical and in-depth reviews of the field point to the different analyses that have already been analyzed for liquid biopsy purposes, circulating tumor cells, ctDNA, or circulating tumor DNA, exosomes, and so on. And the medical as well as scientific popular press is now populated, it has been populated for the last couple of years or more with really optimistic and predictions as to the changing role and changing entry of liquid biopsy into oncology practices. So these are some of the titles of papers that have recently come out proclaiming that, in fact, this is the subject of today, that this is actually happening. And then there are, of course, more sort of original or data-based papers that really solidify the view that, in fact, by applying liquid biopsy approaches, this whole early prediction based on some of the analyses that I mentioned, one can actually change the trajectory of disease and impact in a very significant way patient survival. This is really happening in the clinic right now. And basically, perhaps the most, I suppose, compelling aspect of all this is that liquid biopsy approaches have already entered the regulatory space in a form of different FDA-approved assays that have really, been validated for patient use. And for example, for ctDNA, they include 360 CDX or Foundation One Liquid CDX or MSK Access. And there are, of course, assays, long-standing assay for circulating tumor cells, cell search. And in fact, there's already 3 platforms based the so-called exosomes, to which I'll get into describing a little later. So the question might be, how did it come about, and how come we didn't think of it earlier? How is it that we haven't seen it coming? And we did see this earlier, in a sense, in a collective sense, we as a scientific community. And in fact, it may be worth reflecting for a minute or two on where does this idea and the finding come from. And in fact, they have a fairly long history. So the fact that tumor cells perturb the systemic circulation in very consequential and in some cases diagnostic ways has been known since the 1980s or 1800s rather. There are known reports of coagulopathy in cancer patients, or observation that cancer cells can sometimes be detected in circulation of patients. And then, of course, in 1940s, circulating DNA was first detected, and then it was found that in cancer patients, the circulating tumor-free DNA is actually elevated in cancer patients. And then on, there were findings piling up, so to speak, or accumulating, pointing in the direction that, in fact, biofluids or compartments in the body that are far remote from where the original tumor is can be used as sources of tumor-related material. For example, the DNA methylation could be detected in blood of cancer patients. That in early 2010s, it was found that, for example, some of the oncogenic mutations, such as RAS, can be found in circulating tumor DNA in blood, and so on and so forth. And finally, in recent years, there's been an avalanche of these regulatory approvals. For example, Guardant 360 was approved in around 2020, and then this was followed by other methods, such as, for example, DNA-seq from Sloan Kettering platform. The very term of liquid biopsy was actually coined by Ali Panenvire and Klaus Pantel, specifically for circulating tumor cells, but this term is now widely used to describe all these different analytes and approaches that ultimately lead to this remote detection of cancer by sampling biofluids. So this is the global picture of liquid biopsy, and I wanted to paint this because, although I'm going to speak about pediatric questions more specifically, I think it's important to have a perspective where do we stand in this whole conceptual realm, and how does this relate to pediatrics. And so where do we stand on liquid biopsy in pediatric oncology, the question might be. And here, I just wanted to quote this sort of, movie quote from the movie, which essentially sends the signal that we actually may have a problem. So pediatrics, I mean, this could be followed by, let's say, certain measures in the literature. For example, one of the measures is the number of publications that come out in different fields. And for example, for adult liquid biopsy, we have reached this really dramatic numbers of over 1,500 publications a year. So this suggests there's tremendous intensity of this research, and there's great international, worldwide interest in studying different liquid biopsy platforms and technologies and approaches and so on. And by comparison, pediatric liquid biopsy has barely registered on this publication scale. In fact, in a recent review paper by Lack, it was found that, especially for exosomes, there's only been about 15 relevant studies that were identified, which would contain, let's say, clinically informative material or attempts to extract information from clinical material in pediatric cancer patients. So I would argue that there's this disconnect between the general interest in liquid biopsy as a emerging conceptual breakthrough and technological new technological approach to cancer diagnostic, and what is actually happening in pediatrics. And And this is even emphasized even stronger when one looks at the FDA approval rates, which, as I mentioned, in adult oncology has already been ongoing for some years. And in pediatric, there's currently, to my knowledge, no pediatric biopsy test available at the present time. So then the question is, will there be an upsurge of liquid biopsy application in pediatric cancer? Maybe, but, and this is the second problem. The second problem is that when one looks at the activity of clinical trial projects as recorded by clinicaltrials.gov website, this is maybe a year old, but it's still fairly accurate. When one compares the adult clinical trials, for example, depicted here in yellow, one sees many, often hundreds, of clinical trials ongoing in different aspects of liquid biopsy when search for different search terms. And the purple color here depicts the corresponding pediatric clinical trials. And as you can see, there's only a handful of those. And this leads us to a somewhat pessimistic, or I should say, mobilizing sort of suggestions is that given the relatively few clinical trials, there may not be an immediate upsurge in pediatric liquid biopsy in the near future unless significant investments are made. And I think this is a critical point because it may well be that pediatric cancers are not as frequent and they present considerable challenges in studies on liquid biopsy and beyond, but it is important to invest in this area just to bring this on par with where the adult oncology currently is. So does this mean what I just said, that there's no activity or no interest in pediatric liquid biopsy sphere? And I didn't mean to say that. I think, in fact, there's been a considerable activity over the last several years. And this is depicted by the summary from the recent review paper, where several different disease entities have been studied for different possible biomarkers that may have diagnostic significance for these diseases. For example, neuroblastoma has been a frequent subject of studies with different targets, such as MECAN, for example, or copy number variations or notch, and so on and so forth, listed here. Ewing sarcoma has also been studied for a number of years, and so is osteosarcoma, Wilms tumor, hepatoblastoma, and CNS tumors. In fact, recent developments in brain tumors, such as diffuse gliomas, for example, have been quickly followed by attempts to isolate and analyze ctDNA. This is all ctDNA, signifying the associated mutations, such as, for example, K27M histone 3 mutations, or PDGF receptor A and ATRX, and so on, IDH1. So there's been a considerable level of activity in the ctDNA field, and this is ongoing. And perhaps it makes sense to point out some of the specific publications or successes that really may pave the way forward. So for example, this is a high-profile paper that just came out a couple of years ago from the St. Jude's Center, where these investigators, led by Paul Northcott, have really studied or explored approaches to one particular type of tumor, which is the pediatric medulloblastoma, by taking serial samples of lumbar punctures, or cerebral spinal fluid from lumbar punctures. They were taken not once, but in a serial fashion. And this is an important aspect of this study, because the investigators believe that a snapshot liquid biopsy may actually not really give a full picture of the disease and dynamic. And this is a very informative study in a certain way, because by sequencing of these samples of tumor-derived DNA, these investigators were able to establish that there's certain feasibility of implementing these sorts of diagnostic approaches in this particular brain tumor, in the form of, for example, greater frequency of detection of minimal residual disease, or in anticipating the radiological progression by using by sequencing approach, by showing that there are genetic events preceding the radiological changes. There was also a certain interesting correlation between early detection of MRD using this particular approach and patients' progression-free survival. And finally, and perhaps most interestingly, these investigators were able to detect new events in the trajectory of the disease by showing that, for example, new sequences emerge as the disease moves forward. And so this is one of these sort of really landmark studies, but not the only one. There's, of course, a more recent study from UCLA where these investigators collected a large cohort of patients for pediatric studies. This is not insignificant. So there are 73 cancers and 19 controls, and they analyzed their different genetic features, copy number aberrations, and some of the targeted mutations. And interestingly, in some of these cases, although this was designed as a feasibility study mostly, but in some of these cases, they were able to find that liquid biopsy detected recurrence 9 months before the clinical diagnosis. So this is a fairly significant signal that, in fact, this strategy is actually could potentially be effective in pediatric patients. And then, of course, there's a fairly well-publicized paper in Cancer Discovery where serial profiling of circulating tumor DNA again highlighted the dynamic evolution of the disease. In this case, it was neuroblastoma and ALK mutation. So there are these studies that really show feasibility, although the pediatric oncology may be more difficult in terms of developing this approach, but there's certain feasibility. And then, of course, there are these. So what do we do with this feasibility? There are these early studies, so far very sort of preliminary, that show what can one do with this kind of information. So, for example, in this recent study which has been published this year, these investigators were trying to resolve the issue of pediatric brainstem tumor. And what they did is they collected CSF and analyzed this material for BRAF mutation. And indeed, they found that there's a BRAF 600E mutation in these particular patients using this methodology. And this had a dramatic effect on the natural history of this patient, because the patient was put on trametinib and dabrafenib. And as you can see from these boxed fields, the disease responded in a very dramatic way. And it was followed up to 12 months after diagnosis, and it was still, in relatively strong remission. So this shows what is the potential of this particular approach in some of these indications. Now, so this is all circulating tumor DNA, and of course, this is the center of gravity of liquid biopsy research today. So if that's the case, why should we be talking about exosomes? So why should we bother with these other analytes? And before I attempt to answer this question, I would like to say that, of course, exosomes is just a term that's widely used, but in fact, in reality, it reflects only one aspect of a very large library of different secreted particles that cancer cells are able to emit. And this family of different particles has grown tremendously over the last 10, 15 years, and includes particles of micron size dimensions and particles that are under 300 nanometers dimension, up to very small, tiny particles that are, 30 or something in that neighborhood, 5 nanometers. And so there's a great heterogeneity of these particles, and they can be visualized using different high-resolution techniques, such as like electron microscopy or super-resolution microscopy, but they can also be analyzed in many other different ways. And what was found is that these particles actually are incredibly complex internally. They contain a variety of different molecules. They contain proteins, lipids, RNA, DNA, and so on, which makes them very uniquely amenable to analytical purposes, simply because they carry information about multitude of features that other analytes simply miss. So, for example, they can carry information about identity, state, activity, and responses of cells. They also reflect cell populations because they're highly diverse, so they cover the diversity of cancer cells really well. and they also can contain information about molecular transformation. So these are some of the comparisons. For example, ctDNA is well established, the techniques are well established, the materials are relatively stable, and so on, but there's very low abundance of ctDNA. Not everybody realizes that tumor DNA is only about 0.1 to 10% of the total cell-free DNA in plasma, so it's not very abundant, and there's no information about phenotype, proteome, and other features. In contrast, vesicles, extracellular vesicles, including exosomes, which are just a subset of those, are highly abundant. There's about 10 to the 9 to 10 to the 10th of them per ml of plasma. They contain these multi-analyte entities, RNA and proteins, and so on. They're highly heterogeneous, stable, and functional, as I mentioned earlier. Now, there's only one problem with vesicles or nanoparticles as analytes, and that is how to analyze them. And so they pose certain challenges because they're intrinsically complex, heterogeneous, variable, and also invisible to standard optical microscopy. So if you can imagine, this is a cross-section of the human hair. This is a cell, red blood cell, and this is a size of bacteria. This little dot that you almost cannot see is the size of an exosome or small extracellular vesicles. So how can one study something that one cannot see, and at least not see using standard microscopy approaches? So to answer this question, at our institution, we developed a technology platform that is equipped with instruments specialized to either isolate, quantify, phenotype, and sort these particles. And with these instruments, we can obtain data that goes beyond what standard techniques can do, and learn about these particles in far greater depth than it would have been possible using non-specialized or journal methods. And with this material, we moved on to some new technologies. For example, we collaborate with bioengineers at McGill University who developed the non-commercial strategies, for example, one of which is the Raman spectroscopy analysis, where Professor Shah Makshid and Marcel Jalaik have developed this platform where what is called Raman effect is detected in single extracellular vesicles trapped in these cavities, and basically the Raman effect is simply the energy emission after certain chemical bonds have been excited with laser light, and you can collect this information in the form of this Raman spectra, subject this to artificial intelligence processing, and bingo, you can basically use plasma circulating vesicles to separate patients who do not have any clinically apparent disease from patients who actually are diagnosed with glioblastoma. So there's certain technological solution to the problem or to the challenge of extracellular vesicle analysis, and those are emerging and evolving as we speak. So could these technologies then help us to understand and develop new approaches to liquid biopsy in pediatric oncology? And we like to believe that the answer is yes. So at our institution, we formed this research group called NET, which stands for Nanomedicine Program in Exosome Liquid Biopsy in Children with Cancer. And this is essentially the composition of this group, some of the investigators and their teams, and their sole purpose is to simply use some of these technologies and approaches to adapt or rediscover pediatric approaches effective in pediatric liquid biopsy applications. Of course, we are not the only ones doing this, and one can argue that if extracellular vesicles or EV-based liquid biopsy in pediatric cancer is actually gaining momentum internationally. So these are the excerpts from some of the review papers that have recently come out, and there are efforts to focus on different types of analytes in different disease entities such as brain tumors, lymphoma, hepatoblastoma, neuroblastoma, and so on, and focusing primarily on some of the microRNAs or proteins that can be detected in these vesicles in different biofluids, plasma or CSF in some cases. And this is just a summation, a table summarizing some of these efforts. And so, for example, for different disease states, just like in the case of ctDNA, one can actually detect a cargo, for example, such as mRNA for in neuroblastoma using extracellular vesicles as carriers. And they have several advantages. For example, they're highly stable. They protect the RNA from degradation. They allow multiplexing, so one can look at multiple analytes at the same time in a single vesicle setting, and one can detect all these different properties in the process. So there's a handful of papers that came out, not very many, but the field is evolving. There are others who take a completely different approaches. For example, by mining the data from single cell and multi-omics analysis. For a group at German Cancer Research Center has come to realization that some of the signatures of patients of medulloblastoma samples that they've analyzed in their patients actually have a particular configuration of genes which suggests that these tumor cells may produce a very particular type of extracellular vesicles, the vesicles that are called large oncosomes, based on the composition of these proteins. And they use this tool to analyze the survival of these patients using two different cohorts, and they found that the presence of the signature of this is not a direct measurement of vesicles, but it's an analysis of the potential of tumor cells to produce these large oncosome-like vesicles. This was found to be predictive of a poor survival. So basically, this suggests that this particular property of cancer cells may in fact have prognostic significance. So what would make, other than technicalities, what would make extracellular vesicles attractive, and why would one want to study them as a liquid biopsy analysis platform? I would argue that it's not just their analytical properties, some of which were mentioned. So there are numerous, they're complex, they can reflect tumor subpopulations accurately, but also the fact that unlike ctDNA, for example, extracellular vesicles actually have a biological or functional role in cancer. So by detecting them and their properties, one can shed some light on what do they actually do in cancer and associate this with certain clinical features that might be of interest. I would like to illustrate this by example of some of the experimental studies we have done some years ago, which involved adult brain tumors, but where the paradigms or the basic principles could be easily transplanted or translated into pediatric oncology. So these studies were actually initiated by our interest in some of the oncogenic molecules, such as epidermal growth factor variant 3, which is a known oncogene in human glioblastoma. And some of my colleagues in the laboratory have been studying different cell lines that express this oncogene or don't express it. And what they found, serendipitously somewhat, is that when one isolates conditioned medium from these different cells, or the material that the cells secrete into the fluid in which they are cultured, this material actually did contain, surprisingly and unexpectedly to us, they contain the very same oncogenic proteins that we find in cancer cells themselves. And this was a surprising finding, because we were always taught that oncogenes are basically attached to cancer cells. The oncogenic mutations lead to oncogenic proteins, and this is all happening within the confines of cancer cells. Here we had these oncogenes exteriorized, as it were, to the exterior of cancer cells. And we couldn't believe it, so we confirmed this with using some of the more advanced technologies. In this case, this is nanoflow cytometry. They can detect these nano-size particles, and we find that there's a subset of these particles that actually contain this oncogenic EGF receptor variant 3. And then we will wonder whether or not this is actually going to be detectable in blood of experimental mice with these tumors. And the answer was yes, it was detectable, or in patients with non-experimental, very real glioblastoma. And yes, the answer was a yes again. And there are larger cohorts now that speak to the same point. So of course, this is now all the kind of descriptive analysis, and we were very happy to see that this potentially could rise to the level of a biomarker. But the question was, what do these vesicles and containing oncogenic receptor actually do? And And at some point, we incubated these vesicles with cells that did not have an oncogenic expression of EGF receptor Y3. We found that this expression is actually acquired in a horizontal manner. So vesicles transmit this receptor between cells and basically render the non-transformed cells transformed, at least transiently, by transmission of this oncogenic receptor. So there's a real function behind these vesicles and their cargo. And this function is actually important because the cells that have taken up vesicles containing EGF receptor Y3 acquire certain properties of malignancy, such as increased clonogenicity, as a function of their uptake of these oncogenic vesicles. There are also other examples. For example, in a recent study, a student in my laboratory, Christiana, has been wondering, for example, what would happen if these vesicles containing oncogenic EGF receptor when they come into contact with blood vessels in the tumor. And so she conducted some of the related experiments, and to make the long story short, we've observed that tumor-derived extracellular vesicles containing oncogenic EGF receptor are grafted, as it were, into endothelial cells in tumors, and the consequence of this is rather dramatic, because tumors in which this process is occurring form a very different set of blood vessels. Their blood vessels are no longer small and angiogenic, but they become dilated and enlarged. And this is a process we call vasectasia. This process is actually strictly dependent upon transfer of this oncogenic EGF receptor to endothelial cells and its activation in this foreign cellular environment. Another example of extracellular vesicles playing a role in brain tumors is encapsulated in another study from this time from Lata Adnani in my group. Lata was very interested in a different type of glioblastoma in which the cells have more neuronal-like properties. They're called proneural glioma stem cells, and these cells maintain their stem-like properties in the presence of neighboring cells that are like them, as well as their derived extracellular vesicles that these cells shed into their milieu. Now, if these cells come into contact with blood vessels, extracellular vesicles from blood vessels actually overpower these effects and alter the phenotype of these glioma stem cells into more mesenchymal type tumors. So there's this mutual reciprocal dialogue mediated by extracellular vesicles between tumor cells and blood vessels. And although I've used the adult glioma as an example or paradigm, these sorts of processes are very likely to occur also in pediatric tumors, and they're worthy of our attention because these vesicles that mediate these effects, they may, of course, be very interesting and molecular mediators and molecular effectors of these effects, but they also could be potential analytes for liquid biopsy approaches. So there's also a bigger picture to it. As the time progresses over the last 15 years or so, the various functional roles of extracellular vesicles have grown enormously and now encompass a great variety of processes, such as degradation of extracellular matrix, metastasis, formation of pre-metastatic niches, immunosuppression, inflammation, vascular permeability, and so on. Even metabolic effects. For example, it was found that extracellular vesicles from cancer cells can alter the insulin production in pancreas and render pancreatic islets hypofunctional or dysfunctional. So there's the point here is that these vesicles that have all these effects, they have biological activities of interest, but they also carry information about these alterations that could be exploited for diagnostic purposes as a liquid biopsy approach. So just a few closing comments. Where is the extracellular vesicle-based liquid biopsy field going, so to speak, or and this is, of course, my personal opinion. I'm not claiming any generality for this, but where would this be going in? What can we expect, or where is the field headed for now? And I would argue that one area of particular interest that is emerging and could impact the pediatric liquid biopsy effort in a very significant way is profiling single vesicle landscape. So these technologies that people have used to profile extracellular vesicles for diagnostic purposes have usually used a collection of vesicles that could be isolated from biofluids in a mass, so to speak, and analyzed using different RNA sequencing or protein seeking approaches. But we have to realize that the heterogeneity of vesicles coming from different cell populations is in itself an informative feature, right? And so by using some of these techniques that I've described earlier, by single vesicle profiling, either in using different probes or using label-free approaches such as Raman spectrometry, one can eventually arrive at collective image of the heterogeneity, or a hologram, as it were, of vesicles from both normal and cancer cells in a biofluid space of interest, and potentially reconstruct some of the cellular contributors to this heterogeneity. And this kind of comprehensive approach could be used for new generation liquid biopsy, in using multiple biofluids, using multiplexing approaches, using monitoring in real time in non-invasive way, and in a way reflective of cellular complexity. So on that note, I'd like to close, because we need some maybe time if there's any interest in discussing some of these things. But in thinking here, I'd like to emphasize that liquid biopsy in pediatric oncology is in early days, but it's coming with force, and it'll very likely and almost certainly change the way patients are treated and monitored, and this approach will very likely change the outcome. So thank you very much for your attention, and appreciate it once again being invited.
Thank you, Dr. Rock. We have one question. Just to remind everyone, you can ask your questions using the Q&A feature. If you would like to ask the question directly, please raise your hand and I can unmute you to ask your question. So one of the questions we have is, can liquid biopsy using exosome as an analyte be used as a newborn screening tool to detect cancer predisposition syndromes?
Yeah, so there is a significant interest in this area. People, for example, there's in fact investigator in our institution who's studying this. So obviously there's an interest in, for example, more following exosomal profiles in syndromes such as Li-Fraumeni, for example, or Beckwith-Wiedemann, and so on. And there's no, to my knowledge, published study with conclusive answers to these questions. For example, one would expect that, for example, in conditions such as, for example, Li-Fraumeni, these patients develop spontaneous tumors due to loss of heterozygosity in p53. That therefore one would expect that as these lesions occur in a clinically silent way, a liquid biopsy measuring, for example, some aspects of this would facilitate, for example, awareness and maybe some in-depth investigation of these patients for potential cancer-like events. So this is a very viable option. This is being discussed and is being studied. I don't think there's any ready-to-use or available pipeline for this at this point, at least not that I know of. And but I think it'll come out fairly soon, I think.
Okay. We have another question. What factors regulate the magnitude of exosome production and circulation, and does this phenomena occur only in malignant tumors?
No, so to take the last question first, no, of course, exosome or extracellular vesicle production is a feature of all cells. This is a very rudimentary property of cells, highly conserved from bacteria to eukaryotes and of course in humans as well. And but the cancer cells impose certain regulatory imperatives on this whole process. So there's multiple pathways of course of vesicle biogenesis. At least two pathways are responsible for exosome production. There's more pathways responsible for production of these other types of extracellular vesicles that emanate from plasma membrane or the and they are regulated by oncogenic pathways and so on. So there's many elements that could influence this process. One of which is, for example, cellular stress. So it is known, for example, that p53 pathway controls. It's been known for the last 15 years or so that p53 pathways controls process of exosome biogenesis, and cells that lose p53, for example, do not respond to radiation, for example, by the increase in production of extracellular vesicles. It's known that certain oncogenes such as RAS and MYC and aurora kinase B have a often dramatic influence on the numbers and the type and the cargo of extracellular vesicles. Hypoxia changes the composition of extracellular vesicles, so cell differentiation impacts vesicle repertoire. And this is a very important question in a sense, because I just breezed through this very quickly, but extracellular vesicles have this unique ability to reflect these states in biofluids. So if it's theoretically possible, although this has not been really documented solidly in the clinical realm, that for example, the level of hypoxia can be actually read out of the cargo of circulating vesicles, because it's known that, let's say, in cell culture systems, subjecting cells to hypoxia changes the cargo of vesicles. There's appearance of hypoxia-regulated gene products in the vesicle cargo, which then can be interpreted, in terms of hypoxia or lack thereof in the tumor. So there's many challenges, but there's also many opportunities that extracellular vesicles carry with them, as I attempted to signal. So yeah, that's a great question, and there's many more. Like almost every process that impacts the originating cells would actually have an impact on vesicle repertoire, one way or the other.
Next question is, what future research plans do you have for your own lab regarding exosomes and liquid biopsy?
Yeah, so no, I have many plans. So our interests are divided between our really passion, I would say, for the liquid biopsy approach, where we formed this multi-investigator group to really tackle this in a significant way. So we would like to develop these sort of pediatric specific assays that, or at least build a foundation for it that would allow us to make these diagnostic inroads. But there's also a fact, as I attempted to convey, there's also the functional component to it. So we are very interested in, for example, what do these vesicles actually do, let's say, and in many pediatric cancers, for example, there are processes that at least they're amenable to vesicle-mediated regulation. So, for example, I mentioned the vascular processes could be altered by vesicles. We are now extending our studies that I mentioned briefly into pediatric realm, and of course, the question being, are extracellular vesicles contributing to the vascular rearrangements that are seen in cancer? That are, of course, extremely important for different therapeutic aspects of care about this patient radiation response and chemotherapy response and so on and so forth. So these are some of the of course, the immunoregulation becomes a very sort of hot topic, and it's of great interest. It's known that extracellular vesicles in adult cancers, for example, in melanoma, have a profound effect on immunoregulation. And of course, the question is whether or not in pediatric cancers similar processes are taking place. And so these are some of the interests that we have in the laboratory, and in the role of extracellular vesicles in metastasis. We collaborate with with a group that has a keen interest in metastatic pediatric cancers, and of course we are trying to help with this study as well. So yes, there's a handful of these topics that we are already pursuing.
Next question is exciting talk, beautiful overview of the liquid biopsy field. What is your perspective on one, sensitivity, and two, ease of clinical implementation for ctDNA analysis compared to EV profiling? And secondly, okay, you can finish.
No, sorry, go ahead. Sorry. Yeah, I wasn't
And
sure if this is.
secondly, do you think there will be added value of simultaneous ctDNA and EV profiling for routine clinical use to enhance sensitivity of detection, inform targeted cellular therapy, etc.?
Yeah, so this is a great question. Again, I think there's one point that I would like to maybe highlight is that they don't, like ctDNA and extracellular vesicles don't have to be two different entities, right? So there was a wave of findings to which we also contributed to some extent, which led to the notion that extracellular vesicles from cancer cells may actually contain genomic DNA, and in fact, genomic DNA associated with extracellular vesicles is actually longer than the soluble ctDNA that one finds in plasma, right? So it may be that some aspects of ctDNA research is actually predicated on processing extracellular vesicles, containing these genomic sequences. Part of this question was also whether or not there's, like, even if this was taken aside, is there a value in analyzing simultaneously extracellular vesicles and ctDNA? I would say absolutely yes. I think as with any other challenging clinical problem, there is no single marker for anything, right? I mean, in the areas where medical progress has been made, let's say, treating diabetes or cardiovascular disease, there's always multiple ways of looking at the different aspects of the disease. And there are certain things that ctDNA could tell us more easily or more readily than with extracellular vesicles, but there's also other areas where ctDNA would completely fail, right? So ctDNA would never tell us whether or not, for example, the tumor has been in hypoxia, for example, or whether the target of drug X has been hit, right? That we may tell. So this is where combinations of these readouts or these platforms, as it were, could be very interesting. And I think this is being recognized and investigated. There, I know of a number of groups who take that approach, that they basically are trying to integrate the liquid biopsy based on extracellular vesicles and ctDNA. There was a component of this question which sort of had to do with feasibility, and is it easier to detect ctDNA or is it easier to detect extracellular vesicles? I would say that it depends what is being detected, because with these novel approaches, for example, I flashed this sort of piece of data that we've obtained with using the Raman spectrometry. I think one needs probably something like 5 microliters of vesicle preparation, which comes from maybe 50 to 100 microliters of biofluid plasma, that can give us a distinctive Raman spectrum, right? And everything is sort of automated and everything is unbiased, so it's all machine learning based approach, and so this could be very sensitive and could be very sort of informative, maybe on par with what today's ctDNA techniques are going to offer. So yet, of course, ctDNA has advantages, and it's an older, more established technique. There's some amazing, advances in, for example, sensitivity, using, for example, the droplet digital PCR, or using, sequencing approaches where this, it could really get very sensitive and accurate. So I don't think these are mutually exclusive. I don't think there's a race between two different competing platforms. It is I think it's more of a convergence. where as we make advances in extracellular vesicles, we open certain questions that would otherwise not be accessible, and yet at the same time, I think the field is perfecting ctDNA technology in a very sort of impressive way, as some of these recent papers have indicated. So I don't know if this answers your question, but I think it's just, I don't think it's a yes or no answer. I think it's a more nuanced approach where most certainly multiple platforms will eventually be integrated, I suspect, or I hope, and that will give us the most satisfying answers.
And one last question. Isolation of EV subpopulations is still an outstanding problem in the EV field. How did you confirm that microvesicles are the only vesicles that carry EGFR?
So this is actually even more complicated than that, because we know that, for example, cancer cells that produce, let's say, certain cargo, let's say EGF receptor, in a form of extracellular vesicle cargo, that they at the same time produce vesicles that don't have EGF receptor, right? So not every vesicle that comes from cancer cells has zero cargo of interest, and that's the way it is. This is the biology of this process, and I think it's not neither good or bad. It's just the way it is. So basically, there's two ways to approach this problem. Let's say from diagnostic or liquid biopsy perspective. One is to enrich or purify vesicles of interest, and of course this could be done. There are immunoaffinity platforms that could be deployed to, for example, purify vesicles that contain mutant EGF receptor, for example, and this could be used to solidify these findings. But there's also, as I attempted to propose in my last few comments, there's also an alternative approach, which is sort of an approach which takes a more panoramic view at the whole process, whereby we wouldn't want to focus on one or two specific targets, but we would want to reconstruct the landscape of vesicles from not just cancer cells, but cancer cells and inflammatory cells and the stromal components as it changes with specific disease states. And I would argue that this more comprehensive landscape that could be achieved, by using, there are platforms now that can detect, for example, 15, 20 or more analytes per vesicle, right? So there are microfluidic devices. Even here at McGill, people make these things that every vesicle can be probed for 15 different proteins, right? And so this gives the resolution. So my inspiration in this regard is, for example, what has happened with single cell sequencing vis-a-vis traditional sequencing in the space of tumor cell analysis. I feel that single cell sequencing has added this component of heterogeneity that was previously never accessible, and I think the same could happen eventually with vesicles, with the progress in these technologies, which may eventually give us this more panoramic, more multi-analyte and heterogeneous perspective on how the vesicle landscape changes with disease, and how we can read from it what needs to be done and what kind of interventions would be most effective. So I think this is, but of course it's valid to say that for now, yes, there's an interest in purifying vesicles with specific oncogenic cargoes, and there are ways to do it, and there's ways to validate this and confirm the identity of these vesicles.